Hi, I'm Louis-Pierre Como, soil carbon scientist for the federal government of Canada. Today I will summarize the minimally disturbed microcosm core method to assess soil respiration in a lab. This method consists of 7 main steps. First, collecting the soil sample. Second, pooling the soil samples per treatment. Third, removing the visible roots. Fourth, repacking the soil samples into microcosms. Fifth, incubating the soils per experiment. 6. Assessing the gas concentration, and 7. Calculating the gas fluxes. The soil sample can be collected with intact cores or any other tools. If they are collected with intact cores, then they would have to be pulled in the lab afterward. And if they are collected with other tools, they can be pulled directly in the fields while collecting the samples. A paper filter needs to be placed at the bottom of the microcosms to prevent the soil and the water to get out the microcosms cores from the holes that are below. That paper filter needs to be weighed in dry and in wet to know the exact weight of it with the microcosms, the empty microcosms. So later on we can assess and adjust the water concentration, the moisture content in the microcosm cores. It is important to remove the visible roots and plant fragments that we can see in the soil samples before repacking the soil into the microcosms, and to carefully homogenize the soil together without causing too much disturbance in it. The amount of soil to be put in each microcosms will depend on the moisture of the soil samples and the aimed bulk density to be fixed in the microcosms. To ensure the bulk density is homogeneous in the microcosms, the repacking is made in 2 steps. One repacking at 2.5 centimeters and the second repacking at the line of 5 centimeters. Depending on the aimed moisture content, a few drops of water are added to the microcosms, or the microcosms could be air dry for a few hours. To prevent moisture loss during the pre-incubation, parafilm is being put on the top of the microcosms during the pre-incubation step. The 2 weeks pre-incubation period should be done at the same temperature as the main incubation that will follow. It is important not to breathe inside the container during the box closure and to ventilate it a little bit to prevent artificial time zero CO2. The incubation can be done at different temperature, and 25 degrees generally considered as a standard. The gas sample can be collected at a different times interval, but time 0, 24 hours, 48 hours, and 72 hours are generally considered standard to produce the regression lines. Before collecting the gas sample, it's important to make a small gas flow inside the containers to collect the homogeneous gas sample from the incubation box. Normally 20 mils of gas from the incubation box will be sucked out and put into excitaners for future GC analysis. That GC analysis should be done within 48 hours of gas collection. This 5-minute presentation was a summary of the methods, and many details could not be explained. All the details about the methods were published in our main paper in 2018 and Method X, and other papers too that compared the method with other standard procedures were recently published. For any further questions, you can send me an email at my email address that is stated here. Thank you very much.